I'd like to invite Dr. Srikanta Goswami, who is going to talk about functional characterization of long non-coding RNAs in pediatric pancreatic ductal adenocarcinoma.
Thank you, Chairperson. At the outset, I would like to thank Dr. Sudipta Roy Choudhury and his team for the kind invitation. And also it's an honor for me to share the session with Professor Whitcomb. Actually, I learned about the disease reading his papers and reviews. Thank you, sir. So, pancreatic cancer doesn't need any introduction to this audience. And like chronic inflammation progressing to pancreatic cancer is really a hot topic. And we call ourselves molecular gastroenterology laboratory. And the main focus of our lab is actually like looking at the trajectory of chronic inflammation progressing to malignancy through multiple different aspects. And being a institute of biomedical genomics, we definitely use to try to follow it's like a reverse pyramid approach, trying to look at the global changes and then integrate those results and then functionalize those leads so that like you can have a causation. So the association or correlation to causation, that we like to focus. And definitely one arm of my lab works on if those leads could be of use of any diagnostic biomarker. So the story I'll share with you today is just recently developed stories on non-coding RNAs. So long non-coding RNAs, to be more precise, and long non-coding RNAs are actually non-protein coding genes which are more than 200 nucleotides. And if we look at the functions, they function at multiple levels, starting from regulating the transcription, then RNA processing, processing of other mRNAs, and then translation, and they also act as decoys of microRNAs. So microRNAs are also gene regulatory molecules. They act as decoys of microRNAs so that other mRNAs, expression of other mRNAs are more. Very recently, some of those long non-coding RNAs have been also linked to the production of small peptides. So despite discrete candidate long non-coding RNAs, not much was known for global changes, what happens to the global change of long non-coding RNAs in pancreatic cancer. And we performed a microarray analysis of about adjacent normal pancreatic tissues and pancreatic cancer tissues. And we derived a set of deregulated long non-coding RNAs, both upregulated and downregulated. And then we matched with the TCGA data, that is the cancer genome atlas data of pancreatic cancer patients. And then when we wanted to see the common ones, we found a significant number of long non-coding RNAs deregulated. And very interestingly, that among the top 15, there came 3 different long non-coding RNAs, CAC19, CC81 and PVT1. The importance of these 3 long non-coding RNAs are they reside in the same genetic locus of 8q24-21 region. For the cancer biology is very important because this is one of the mutational hotspot region. So we are very excited why all of a sudden the 3 long non-coding RNAs among the top 15 would be there in the same region. Definitely they might be having some common regulatory phenomena. So one of my students, Momita, she continued with the CAC19-1, which is actually a 324 nucleotide long non-coding RNA, but not much information was available in many cancer apart from few reports. The first thing we did is checked by RT-PCR in our patient cohort, and we found a significant increase in the expression level as comparable to TCGA. And then we checked in the available pancreatic cancer cell lines, and it was really elevated, and the metastatic, the ASPC1 has a most increased expression. And then we cloned the gene and then did the subsequent overexpression and knockdown construct. We developed shRNA against that to actually interrogate the function. Then we conducted a series of assays to interrogate the oncogenic potential of the long non-coding RNAs. Firstly, we performed the MTT assay, where we have found that increased expression of that particular long non-coding RNA increased the survival potential of the cells and vice versa. Then we found high CSC19 expression promotes G1 to S phase transition of the cells. We found a significant increase in G1 to S phase transition when overexpressed, and the siRNA mediated knockdown did the reverse. High CSC19 expression also found to be inhibit apoptosis, and the siRNA mediated knockdown did the reverse. These are the standard assays we need to do before establishing the oncogenic role, and then we did the wanted to find out the role in invasion and migration, perform the wound healing assay, and the overexpression actually facilitated the wound healing, and siRNA mediated knockdown did the reverse. Then we performed the migration and invasion assays and also found the similar results. So then automatically the next step was to find out the EMT marker, check the EMT markers, and the conventional EMT markers we checked. The cadherin was down and the other mesenchymal markers were up. So the most important step for a long non-coding RNA is to find out its location, because the subsequent function depends on where it is actually located. If it is nuclear, the strategy would be different. If it is cytoplasmic, the strategy will be different. And interestingly, we found CSC is almost entirely located in nucleus. So then the obvious step is to find out if CSC19 has any role in transcriptome alteration of the cell. And we did overexpression and knockdown, and we found a significant number of genes were altered when CSC19 expression was perturbed, indicating that it has some role in transcriptome remodeling. And so we did the subsequent pathway analysis, and the EMT or metastasis related pathways emerged to be the most enriched ones. Now, the long non-coding RNAs, how do they function? As you have seen in the first slide, that the non-coding RNAs generally cannot function on their own. They have to bind RNA binding proteins. So the most important part towards exploring its function is to find out what are the RNA binding proteins it interact with. We then transcribe the biotin-related long non-coding RNA, and then subsequently pulled down the RNA binding proteins, and then did a mass spectrometry to find out what are the RNA binding proteins attached with them. So and we found the antisense was a control, and we found 53 RNA binding proteins which exclusively interacted with the same strand of the CSC19. So this is a conventional strategy for this kind of experiments. So we did further tried to shortlist further that 53 RNA binding proteins. There are certain bioinformatic data sets which actually predict what could be the RNA binding proteins binding to them, to the long non-coding RNAs, and we used results from 2 such bioinformatic data sets and combined with one of our earlier published study of RNA binding proteins altered in pancreatic cancer. And after all these combination, we actually landed up in one RNA binding protein that's called the PSPC1, which is actually a paraspeckle component one that's again that's resided totally in the nucleus. It's a nuclear RNA binding protein, and it has been implicated in multiple studies, but not much information, absolutely not much information in pancreatic cancer. So the next set of experiments were actually, I am not going to go, because of lack of time, I am not going to go to details of that. Next set of experiment is to establish the interaction of CSC19 and PSPC1 in vitro in overexpressing CSC19-1 and checking the level there, whether they're directly interacting or not. We did a RNA pull down assay and found the PSPC1 is actually precipitating with the RNA. And interestingly, we found the overexpression of CSC19 is not affecting the level of mRNA level of PSPC1, but rather it's increasing the stability of PSPC1 protein. So that was really interesting. And the obvious experiment is to see do a cyclohexamide chase assay and also use MG132, the proteasome inhibitor, to find out what is happening to the protein degradation. And we found that overexpression of CSC19 actually inhibited the ubiquitination of PSPC1. Again, I'm just running, not explaining the experiments in detail. Then we performed a rescue assay. So what is a rescue assay? We found CSC19 overexpression. Our hypothesis was the function of CSC19 overexpression is mediated through stabilization of PSPC1 and then subsequently the epithelial to mesenchymal transition. So if we use the siRNA against PSPC1, even after overexpression of CSC19, we would not be able to do the subsequent metastasis or so. So that's the rescue assay we performed, and then we also found the interaction of there was a report that PSPC1 interacts with beta-catenin in nucleus and favors in retention of beta-catenin in the nucleus. But what we then wanted to see whether CSC19 has any role or not in this process. And we found increased expression of CSC19, increased the stability of PSPC1, and retention of beta-catenin in nucleus was also retained, also increased. So that actually led to the model where we propose that CSC19, when the level of CSC19 is less, PSPC1 is ubiquitinated and degraded, and the transcription of metastasis gene is less. On the other hand, when CSC19 expression is more, the PSPC1 level is more because of stabilization of the protein, and then it helps in retention of beta-catenin in the nucleus, leading to the transcription of EMT-mediated genes. So I'm happy to share that just last week it got accepted in BMC Molecular Medicine. And so what next? Interestingly, we also wanted to see the interaction, the actual interaction between the two molecules and what are the domains for that. And we found that we actually identified the domain of PSPC1. There's a typical RNA binding domain, RRM, and the interacting domain of long non-coding RNA CSC19. And the next step actually would be to characterize the drugs or small molecules which could inhibit the interaction. Because what we propose for the transcription of metastasis-related gene in pancreatic cancer, the interaction between CSC19 and PSPC1 is necessary. So if we could design a drug for which actually prevents the interaction, that could be key for inhibiting the process. Okay, thank you for your attention. So this is my group, and it's my.
Thank you, Dr. Goswami. This was another wonderful lecture, explaining, getting to know the pathogenesis, and then proposing how to tackle the problem.